Hello everyone, it's Dr. Wu. I'm honored to have this opportunity to introduce my research results to you. And I would like to thank every member of my research team and Science Father for giving me this opportunity to showcase my work. The content I'm going to introduce today is human placental extract regulates polarization of macrophages via IRGM NLRP3 in allergic rhinitis. I will briefly describe the experiment. As we know, AR is a common nasal-mucosal disease. AR is prevalent worldwide, and its pathogenesis is still unclear. The typical symptoms are usually triggered by pollen, mold, dust mites, and animal dander in the air, including sneezing, runny nose, nasal congestion, and nasal itching. AR, which is not only affects their quality of life, but also imposes a heavy social burden. HPE, prepared from the placental of healthy pregnant females, is known to have diverse bioactive and was also reported to have anti-stress effects, improve liver regeneration, and reduce inflammation as well. However, the effect of HPE on AR remains to be explored. Therefore, in the present study, we set up an OVA-induced AR rat model and evaluated the effects of HPE on AR. Different types of cells, including B and T cells, eosinophils, basionophils, and mast cells, are part of an inflammatory process. Macrophages, which are important for our immune system, not only help present antigens, but also play a role in regulating allergic immune responses by producing cytokines and inflammatory substances. Research has shown that macrophages come in different types, like M1 or M2, and the balance between these types is important for the outcome of an organ during inflammation or injury. Another protein called NOD-like receptor protein 3, NLRP3 inflammasome, is a key component of the multi-protein complex and belongs to the NOD-like receptor family. It can be activated by various stimuli, such as RNA viruses, endogenous danger signals, and environmental stimuli. Studies have shown that after NLRP3 activation, both in AR patients and AR mass, caspase-1, IL-1 beta, and IL-18 increase, promoting the progression of AR, while inhibition of NLRP3 can improve nasal inflammation in AR mouse models. Recently, macrophages have been implicated in the contribution of NLRP3-mediated apoptosis to AR development. However, the role of macrophages in AR is still not clear, and the relationship between NLRP3 and macrophages polarization needs further investigation. In this experiment, we established 5 animal model groups using SD rats and the AR model rats were sensitized using OVA. Serum and nasal mucosa tissue were collected at the end of the experiment, and the levels of inflammatory factors in the serum were detected by ELISA. The morphology of the rat nasal mucosa was examined by HE, AB-PAS, GIMSA, and Mason staining, and real-time PCR was used to detect the mRNA expression of NLRP3 and ASC. Western blot immunohistochemistry were used to detect the expression and localization of NLRP3. HPE or polarization treatment greatly reduced both the number and the size of goblet cells in AR rats, and improved the morphological changes of the nasal mucosa in AR rats. Being consistent with the results of Western blot and real-time PCR, we reconfirmed the surprising effect of HPE or polarization on NLRP3, and no significant differences showed between these two groups. Quantitative proteomics detection was performed on the nasal mucosa tissue of rats in the control group, AR model group, and HPE treatment group, and differentially expressed genes were screened by bioinformatics analysis and validated by RT-PCR. Western blot and immunofluorescence double labeling were used to detect the protein expression and localization of IRGM in the nasal mucosa of each group of rats. ELISA was used to detect the IRGM content in the serum of each group of rats, and immunofluorescence double labeling was used to detect the polarization of nasal mucosal macrophages. 53 differentially expressed proteins were screened between the control rats and AR rats, including 22 downregulated proteins and 31 upregulated proteins. In addition, 257 differentially expressed proteins were found between the AR rats and HPE-treated AR rats, including 105 downregulated proteins and 152 upregulated proteins. The expression of IRGM in the nasal mucosa tissue and the content of IRGM in the serum of AR rats were significantly decreased after HPE or polarization treatment. In this experiment, the results show that the optimal concentration for OVA stimulation of NHS cells was 40 mmol per liter. The protein expression of IRGM was concentration and time dependently decreased in OVA-stimulated NHS cells, while HPE or polarization treatment significantly upregulated the protein expression of IRGM. HPE intervention or transfection of the IRGM overexpression plasmid significantly inhibited the high expression of these proteins. In this experiment, we also found that HPE intervention or NLRP3 transfection significantly inhibited the expression of IL-6 and TNF-alpha, but further prompt the expression of IL-10 and ARG1. The results show that HPE significantly alleviated AR symptoms in rats, improved the morphological changes of the nasal mucosa, reduced the levels of inflammatory factors, and inhibited the activation of the NLRP3 inflammasome in AR rats. In addition, HPE treatment reversed the downregulation of IRGM in AR rats, increased the expression of IRGM in macrophages, and decreased the number of IM1 macrophages while increasing the number of IM2 macrophages in the nasomucosal tissue of AR rats. Well, thanks for your attention.